In this video, we'll talk about genomes. We'll talk a little bit about what we have learned from now sequencing thousands of bacterial and archaeal genomes, as well as hundreds of eukaryotic genomes. We will talk primarily about their size, the organization of genes within the genomes, and what's in these genomes. And at the end of this video, you should be able to answer the question, what's in the human genome? Why is it so big? Starting now off with a comparison of genome sizes across a variety of organisms. Here we see bacterial genomes. Archaeal genomes are similar in size. They range up to several millions of base pairs per genome. There's some variation, but that is generally their size. And their genomes tend to be very compact with very little wasted space. When we get to eukaryotes, eukaryotes have larger genomes. There is some overlap between the smallest eukaryotic genomes and the largest bacterial genomes. Not really shown here, but there is. So you can see that when we get to fungi, they just have, in general, larger genomes than bacteria. So that seems pretty nice. Then we get to multicellular organisms. So worms are worms like Caenorhabditis elegans, which are these roundworms that have been used as a genetic model organism and pioneered the human genome sequencing project. And here we see plants, amphibians, and there is mammals. Humans are like other mammals. Their genomes, or our genomes, are 3 billion base pairs per haploid set of chromosomes, or 6 billion base pairs per diploid set of chromosomes. So what we see is that there isn't much variation in range. So compared to fungi, our genomes are enormous. But we're certainly not the king of the hill as far as genome size is concerned. If size mattered, we would be in trouble indeed, because there are amphibians that have much larger genomes. Lots of plants have much larger genomes. And indeed, the biggest genome that we know about is from a single-celled eukaryote, the amoeba. These amoeba have giant cells, and in their nuclei, what we see is that they have somewhere around 500 to 600 billion base pairs of genomes, which would put their genome size somewhere over here on our scale, on our chart. So something for you to consider. We have large genomes, but we certainly don't have the biggest genome. The question then is, what accounts for this variation in genome size? Do larger genomes mean that they have more coding capacity and actually use that coding capacity to make lots more different proteins? Well, thanks to the genome sequencing projects, we can actually look at these genomes in detail and count the protein coding genes. So here we show a chart here from a paper published in 2009 that looks at genome size here on the x-axis, but also then plots the protein coding gene number on the y-axis. And this shows this plot for everything from mitochondria and viruses up through complex eukaryotes. And what we see is that, so here we have bacteria in the white circles, and this is what I mean. The smallest or the largest bacterial genomes are actually a little larger than the smallest eukaryotic genomes. But these really small eukaryotic genomes tend to be obligate intracellular parasites that have reduced genomes and have lost a considerable percentage of their genome. So you can see that there is a wide diversity, a wide variation in eukaryotic genome size. But if you look on the y-axis, there is considerably less diversity in the number of protein coding genes. Figure B shows this in a little different way. It's the percentage of the genome, the percentage of the DNA in the genome that actually is used to encode proteins. And what we see is that as the genome size increases, the percentage of the genome that's used to make proteins actually declines. So that in the largest genome shown here, only about 1% of the genome actually encodes genes, protein coding genes. Humans are somewhere around here. So let's start looking at these genomes in detail, or not in real detail, but more detail than this global picture of genome size and numbers of genes, and look at how genes are organized within the genome. So you should recall that bacteria and archaea, or basically prokaryotes, have one circular chromosome. There are some exceptions, but the vast majority, virtually all bacteria and archaea, their entire genome consists of one circular chromosome. And their major feature is that they're very compact, very little wasted space between genes. So they use most of their genome, a very high percentage of their genome, to encode protein coding genes or otherwise functional ribosomal RNA genes and other types of functional RNA genes. Not only are their genomes very compact so that the space is efficiently utilized or used, what we see is a functional organization into what are called operons. These are units of organization where functionally related genes, meaning genes that encode proteins that all operate, for instance, in the same metabolic pathway, or somehow serve the same function in the cell. They're all grouped together, head to tail, on the same region of the chromosome, right next to each other. The example that I'm going to show you is something called a lac operon. The lac operon encodes 3 protein coding genes, and we'll just call them for now lac Z, Y, and A. And these 3 proteins are needed by the cell to metabolize the disaccharide lactose. In fact, these 3 genes are needed to break lactose into monosaccharides for further metabolism. And the only time the cell actually needs to make these proteins is when there's lactose around. And if there's no lactose around, the cell doesn't need to make these proteins. So it would make sense then for the cell to be able to regulate these genes coordinately. So this is called coordinate regulation. So functionally related genes, the cell will turn them on and turn them off together. Coordinate regulation. To achieve coordinate regulation, what it does is not only puts these 3 genes in the same location on the chromosome, but they're all organized in the same direction, head to tail, as shown here. So the arrow is the direction of transcription. And then they're placed downstream of a single promoter. So recall the promoter is the site where RNA polymerase binds to initiate transcription. So for this operon, there is a single site, a single promoter, for all 3 of these genes. So you don't have to have a separate promoter for each gene, one promoter for all 3 of these genes. This one promoter then directs RNA polymerase to make one messenger RNA. This one messenger RNA has the coding information for all 3 proteins. So there are actually 3 different start codons and 3 different stop codons. So these S's are stop codons. And the AUGs are start codons. So once this messenger RNA is made, RNA polymerases will initiate translation at this first AUG, stop translation at the stop codon, and then reinitiate translation at the next AUG. So ribosomes keep going. They'll first make this LAC-Z protein, then they'll make LAC-Y protein, and then they make LAC-A protein. So this is a very elegant way for bacterial cells and other prokaryotic cells then to achieve coordinate regulation. And I should say that this operon organization of genes is characteristic of prokaryotes, but not in eukaryotes. Now I would like you to think about how bacterial genomes evolve. Given the tremendous diversity of bacteria and archaea and all of the niches that they occupy throughout the planet, from the polar ice caps to the tops of mountains to even miles deep in the bedrock of the Earth. The fact that there's so much diversity among the bacteria and archaea tell us that they have to be able to evolve fairly rapidly. Okay? So how do they do that? Well, we've already seen one of these mechanisms. This is transformation. This is their ability to pick up genetic information from their environment and incorporate it into their own genome. In addition to transformation, we have a second process called conjugation. Conjugation is directed or mediated by plasmids. So plasmids are circular DNA molecules. So they're circular like the chromosome, but they're much smaller than the chromosome. So whereas the chromosome will be more than 1 million base pairs, plasmids are anywhere from a few thousand to tens of thousands of base pairs. They don't approach millions of base pairs. So plasmids are circular DNA molecules, and they contain information, genetic information. They contain genes such as for antibiotic resistance. Pathogenic organisms often contain plasmids that confer pathogenicity. Or virulence genes. So plasmids you can think of as sort of packets of genetic information, and these are transferable. They're transferable from cell to cell through a process called conjugation. So plasmids also contain conjugation genes. So conjugation is a form of bacterial mating, okay, where a cell containing a plasmid will make a copy of a plasmid and transfer it to a cell that doesn't have the plasmid through physical contact. So it's a bacterial form of mating, okay. And then finally, phages. So remember, phage are the viruses that infect bacteria. They're a phage that can actually pick up when they infect a host cell. They break the host cell's chromosome into pieces, and some phage will take some of these pieces of host chromosome and package it in with a phage particle. And when this phage particle exits the lysed host cell and infects a new host cell, it will deliver a fragment from the previous host into the new host. Okay? And that way the new host can acquire genetic information delivered by the phage. So there are these three ways then that bacteria can acquire new genetic information, plus the fact that they have short generation times. So during a day, a bacteria population undergoing exponential growth will replicate their DNA many times, and because of this, what we see is that in a chronologically short time, they can have lots of DNA replications. So each DNA replication means that there is some chance of acquiring mutations. Therefore, mutations arise relatively frequently for bacteria with short generation times. So all of these factors mean that bacterial population can evolve quite quickly in chronological time. Now let's switch to eukaryotic genomes. So you recall that eukaryotic genomes are much larger than prokaryotic genomes, and the human genome at 3 billion base pairs per haploid set of chromosomes is actually on the larger side for eukaryotic genes. For instance, yeasts have their genomes encode only 6,000 genes, and the yeast cell has a much smaller genome. So the yeast cell genome is about 1% of the size of the human genome. Okay? Does that mean that human genome encodes 100 times more proteins? As we said before, that's not the case. Okay? What instead happens is that a much smaller fraction of the human genome is used to make proteins. What we see then is that here in red is a representation of the fraction of the human genome that actually encodes protein, the amino acids in proteins. And that's less than 2%. So less than 2% of human DNA encodes protein. Huh. Right. Well, what about the introns? So remember, human genes, most of them have introns, these intervening non-coding sequences, but these introns are transcribed, and so they should be included as part of human genes. So in fact, we see that introns add considerably to the portion of DNA that is used to encode genes. Introns are about 25% of human DNA. So if we include both exons and introns, okay, so remember the protein coding region is called the exons, so exons plus introns together still constitute less than 30%, about 27% of the human genome. What we see is that by far the largest fraction of the human genome consists of these sequences mysteriously named lines, signs, retroviral-like elements, and DNA-only transposons. All of these DNAs belong to the category of transposable elements. This is a transposable elements. These are a fascinating category of DNA sequences. What they do is you can think of them as selfish or infectious DNAs. Okay? So these DNA sequence elements can invade a host cell's genome. They will direct the host cell nucleus then to make more copies of a transposable element. These copies will then insert themselves into other locations. And therefore, starting with one copy of a transposable element, one can then have thousands, tens of thousands, even hundreds of thousands of copies spreading throughout the genome. And if this happens in the germline, then this becomes a permanent fixture in the species genome. And indeed, that is indeed what has happened in the human genome. What we see is that all of these transposable elements, the human genome, during its evolution, or the ancestors of humans, experienced a number of transposable element insertions, which expanded and takes up now 45% of human DNA. And then there are some other sequences that are repetitive sequences, sequences between genes, and some heterochromatin that has not been sequenced. So when we count up all of the protein coding genes, what we see is that there's between 20,000 to 21,000 protein coding genes. And these protein coding genes actually occupy less than 2% of human DNA. Just to give you an idea of sort of the inefficiency with which our protein coding genes use our DNA, this is a typical human gene, and I chose as an example the gene for CFTR, the cystic fibrosis transmembrane regulator gene. Remember, mutations that eliminate function of this gene cause cystic fibrosis. This is on chromosome 7, so this is a stretch of chromosome 7. These numbers represent the first nucleotide and the last nucleotide that is transcribed to make the CFTR protein on human chromosome 7. Yeah. So if you subtract one number from the other, then you get this number, 189,498 base pairs, which is the length of the pre-mRNA before it undergoes splicing. After this pre-mRNA is spliced and capped and poly-A tailed, without the poly-A tail, it still has 6,129 nucleotides. So compare these two numbers, and you can see that most of this gene consists of introns. And then that's exactly what this figure shows you. These lines are introns. These upticks are the exons. So you can see that the horizontal space occupied by these uptick exons is much smaller than the space occupied by introns. So the introns are much larger than the exons. And then the 6,000 nucleotide messenger RNA is translated to a protein of 1,480 amino acids. So if you calculate how much DNA is actually required at a minimum to make this protein, we come up with only about 400 or less than 4,500 nucleotides of DNA would be necessary to make the protein, but instead the cell uses, our cells use almost 190,000 base pairs of DNA. So you can think about why that is, and there really aren't any good answers at this point. So I told you that the human genome has only a little over 20,000 protein coding genes. But that is a really misleading statistic, because what we see is that almost all human genes, and the latest figure that I've heard is about 90% of human genes, are known to undergo what we call alternative splicing. What this means is that if you have a gene, there is one pre-mRNA that is made. So here's our gene, which is the DNA. Okay. And then this pre-mRNA is made. And then it undergoes splicing. Okay. So here's an example of the mouse gene, hyaluronidase 3. And we know that this mouse gene can be spliced in 3 different ways. So the same pre-messenger RNA under different conditions, and maybe sometimes even in the same cell under different conditions, can be spliced 3 different ways. One way, it makes this protein. It undergoes a single splicing, okay, and it makes this protein, okay, which turns out to be the longest version of this protein. A second contains that same splicing, but then a second splicing event removes a portion of this first exon. So there's a hidden splicing site that in the first exon that can be used, and this gives you our middle or our intermediate length protein. And then there's a third version which skips all of this information. It uses this splice site and that splice site, joins them together, and gives you the shortest version of the protein. So all three proteins will have the same N-terminus and the same carboxy terminus, but they will differ in the middle. So here's an example where we have a single gene making at least 3 different proteins. And since we haven't been able to complete yet an exhaustive survey of all of the different splice variants that are possible for every single human gene, we don't actually have an answer at this time of exactly how many different human proteins our cells can make. Previously, we asked about mechanisms that enable rapid evolution of prokaryotic genomes. While eukaryotic genomes, one can argue over evolutionary timescales, can evolve maybe even faster. Okay? So you can, if you think about the tremendous diversity of multicellular eukaryotic organisms, from plants to animals, and that has all happened in the last 600 to 700 million years, whereas prokaryotes have been evolving for, let's say, 4 billion years, you have to say that one has to conclude that eukaryotes have to be able to evolve quite rapidly as well. So what are these mechanisms that facilitate evolution of eukaryotic genomes? Again, we've already discussed one of them. This is sexual reproduction. So recombination during meiosis generates new chromosomal combinations, and that is a tremendous driver of diversity and therefore a feeder of evolution. But what generates new material? Well, an intriguing hypothesis is that it's transposable elements that play a key or perhaps an even essential role in rapid innovation in eukaryotic genomes. Why? Because transposable elements are present in multiple copies, hundreds, thousands, tens of thousands per genome. So when you have the copies of the same sequence, then you can get recombination. Between transposable elements. And this recombination, when the transposable elements are located on different chromosomes, or if you have multiple copies of the transposable element in the same chromosome, but now the transposable element sequences are undergoing recombination with each other, this causes lots of chromosomal rearrangements. So you get deletions, duplications, translocations, pieces of one chromosome being joined to pieces of a different chromosome, inversions, a piece of a chromosome being flipped with respect to the original parental version. And all of these chromosomal rearrangements then can accelerate evolution because they're generating new variants. And so they're actually mutational events. In particular, duplications means that now we have extra genetic material that is now free to evolve and acquire new functions. And then transposable elements themselves are adding new DNA to the genome, the transposable, sometimes the genes within the transposable element can be co-opted by the host for a host cell function. And transposable elements also influence or affect regulation of nearby genes. So the insertion of a transposable element into the regulatory sequence of a eukaryotic gene may mean that the eukaryotic gene is turned off or even turned on at different times. So all of these can be drivers of evolution of eukaryotic genomes. Here is a chart that I would hope that you use to organize the information that we have presented so far, or we have discussed so far, about various aspects of genomics, doing comparisons between prokaryotes and eukaryotes. And I'm not going to take the time in this video to go through this, but I would urge you to, I offer this to you as a way to help organize your knowledge. I want to close by talking about the human genome in sort of global terms. We know that our closest cousins are the chimpanzees and bonobos, with whom we share 99% nucleotide sequence identity. If we include duplications, then we can add another 3% difference. This means that 3% of DNA has been duplicated in chimpanzees but not in humans, or duplicated in humans but not in chimpanzees. But overall, the sequence identity between chimpanzees and humans, or bonobos and humans, is 99%. What about from human to human? As long as we're talking about Homo sapiens, if we just pick two people at random, unrelated, from anywhere on the earth and compare their genome sequences, we will see that they're 99.9% identical. Wow, that's very close identity. But that 0.1% difference gives us all of our individual characteristics. 0.1% at first doesn't seem like a lot, but we have to multiply that by 6 billion base pairs. This gives us 6 million base pairs. So each person varies by about 6 million base pairs from any other person on the planet. And I think you will agree that this is plenty of variation. So this is all variation at the level of single nucleotide changes, differences. In addition, the percentage of difference increases if we include insertions and deletions, okay? And if we include copy number variation, meaning that some DNA sequences are present in multiple copies, and the number of copies varies from individual to individual. So when we include all of that, then we get even more than 6 million base pairs that